Well, thank you, Jim, for such a very kind and heartwarming introduction. And thank you to the organizers who have awarded me this prize today. And I want to say that this is a really special award for me because C.C. Wang was my mentor as a graduate student. And to be able to come back sort of full circle to have received this award is really a fantastic honor for me and a really big privilege. And thank you, everybody, for also coming to attend our symposium today. And I think you'll have a real treat here with the speakers that we have to talk to you today. So the focus of the session today really is I wanted to highlight the biology of the parasites that we work on as it relates to drug discovery efforts, and so how we exploit or even identify vulnerable biology in the parasites to start drug discovery programs. And we'll do I'll be focusing on two different targets in my talk, and also in some chemistry, and then we'll be moving on to various other biological discovery in other organisms. So just to give you a little bit of background on the Plasmodium parasite, malaria is caused by the Plasmodium species, and it's a single-cell eukaryote from the apicomplex and phyla. 50% of the world population is still at risk for malaria, and that represents over 90 countries where malaria is still endemic. 250 million people infected yearly, with about 0.6 million deaths still occurring yearly. 89% of these deaths occur in Africa, mostly in children under 5. And the fact that we still have this disease so prevalent across the world really is a reflection of the fact that drug resistance is always almost a step ahead of us throughout history, in that we've had many therapeutic agents and we've lost almost all of them to drug resistance. And at the same time, vaccine development has been very problematic, and while there is now a vaccine that is used for children, it's only partially efficacious and really sort of benefits from co-administration with drugs. So we're dealing with a parasite that's very adept at developing resistance, yet really causing a lot of mortality and morbidity in the world. So in terms of trying to discover new drugs for this parasite, it's important to know a little bit about the life cycle and some of the complexities. So there are 5 species of Plasmodium that cause malaria. The 2 largest players here are Plasmodium falciparum, which causes almost all of the deaths, and then second, Plasmodium vivax, which has probably the largest global impact, and it also has a latent liver stage that can come back, up to 5 years later after you've been in an endemic area if you're not treated properly. The parasite enters into the human host through the bite of a mosquito. The first stop is it invades liver cells, where it then transforms to these little merozoites. You can see replicates and then ruptures from the liver, and then goes and enters into the red cell stage, which is the symptomatic stage of the disease, where it sets up shop, invading, growing, dividing, and then erupting from red cells, creating this cycle of destruction in our red cells that then leads to the symptomatic stage of the disease. And occasionally, some of these parasites will transform to the sexual gametocyte stage, which are taken back up by the insect host, and then go on to infect another person. And so if we're wanting to intervene on this cycle, we're absolutely going to have to be able to treat the symptomatic stage to cure patients. But liver stage active compounds will be prophylactic, and gametocyte active compounds will provide the potential to be transmission blocking, which could help us prevent resistance from spreading in the field. So currently, the frontline treatment are artemisinin combination therapies. They've been very effective and credited with a big decline in malaria deaths. And we also have a lot of help from insecticides and bed nets in the field, and some limited aid by vaccines. But despite the success of artemisinin, drug resistance has emerged first in Southeast Asia and now in Africa to these artemisinin combination therapies, paralleling the loss of these key drugs through history. And so we really are on the precipice of a potential loss of the current drugs, and then really a big increase in malaria deaths again. So our therapeutic goals really are to identify novel targets so that we won't share resistance with something that's already in the field, and to use drug combinations to help slow down resistance, to try to target all stages if we can within that combination, obviously safe and inexpensive, and with potentially a high barrier to resistance being something else that can help. So I'm going to talk to you today about sort of the mechanism that the field has used to discover new drugs in malaria. And I would say that we've really grounded our attempts to identify new targets and new biology in chemistry. And the reason for that is that if it's not enough for a target, a protein, to be essential for an organism if we're going to use it to discover new drugs, because there's been many examples in the bacterial field where essential is not sufficient to lead to a drug. You really need also to identify druggable targets that bind small molecules with good affinity and that have good properties for in vivo efficacy. And so I'm going to tell you two stories today, one based on a phenotypic screen where we're looking to just find compounds that kill parasites in the red cell stage in vitro assays, and then work backwards to the target. And there we obviously have something druggable from the start because we're able to really impact the parasite. And then a target-based screen where then in these approaches we piggyback off what's already known in other fields to look at targets that were known to be druggable as a possible inroad for new anti-malarial drugs. And so what I'm showing you here is the drug discovery process where we have some kind of screening assay. We then use a chemical library to identify initial chemical matter to take through to the drug discovery pipeline. And then we have an iterative lead optimization program where we're not only going to need to optimize the potency of the compounds, but the properties that drive good in vivo efficacy. And that means we can't have the liver metabolizing the compounds too quickly, so we have in vitro assays to look at both in microsomes and hepatocytes how fast is a compound likely to be metabolized, and then that can help predict whether we'll have sustained plasma exposure once we get to animals, first rats and mice, and then eventually humans. And through this process, we try to optimize properties. Often you optimize one and break others, and that's why it's really an iterative process coming back. Eventually, you'll get to a late lead, and then a drug candidate, and then enter preclinical and clinical development, and hopefully you get to a drug. So our target product profile is we're really looking for either single-dose cures, oral, or monthly oral chemopreventative, or a long-acting injectable, which we heard about very nicely by Wes yesterday in terms of the HIV profiles. There's a very high bar for the pharmacokinetic properties in this drug discovery era because we're looking really for long half-life compounds with human half-lives of over 120 hours to make these objectives. And then, as I said before, multi-drug combinations to reduce resistance. So I'm going to start out with a short vignette on a phenotypic screen that we did. And here I've changed a little bit the sort of pathway to developing the drug and adding in target ID into this because if we're starting with a phenotypic screen, we're not going to know how it's killing the parasites. So we initially screened 100,000 kilodalton library, 100,000 compound library at UT Southwestern. And from this screen, we prioritized 20 scaffolds to validate those hits and then to start prioritizing which ones we might take into an actual chemistry program. From there, we discovered this little piperidine carbaxaminide series that we call the SW042 series here. And it had nice starting properties of a potency of about 190 nanomolar on the parasite, which I'm labeling here as PF3D7 cells, which is a drug-sensitive cell line. And it also was active against PFDD2, which is a chloroquine-resistant cell line that we use in the lab. Had good selectivity against mammalian cell line, HepG2 cells, and initial good starting properties in terms of good solubility and a reasonably low rate of metabolism in these in vitro assays that I described to you at the beginning. So we decided to focus on this target for our lead optimization, and we also thought that we would, at the same time, in parallel to the chemistry effort, work to identify the target of this series. So just a brief little bit about some of the initial chemistry that was done. My colleague Joe Reedy was pretty rapidly able to move the compound properties towards a better profile. So on the top, I have the target values. We're looking to be on less than 10 nanomolar on the parasite, to have this clearance value in these in vitro microsome assays at less than 3 to give a good prediction of good animal exposure once we dose in animals, and to have really good selectivity and solubility. So you can see starting from the initial hit at 90 nanomolars, we were able to pretty quickly identify a 3 nanomolar compound in this SW584. It still was reasonably metabolically stable and had good selectivity still against this human jerkit line and also really good solubility. So the program was able to identify low nanomolar compounds that were selective and soluble. We fully profiled this compound, which I've accidentally changed its compound name here, SW584/18-192, until it met early lead criteria. At this point, we could tell that the human half-life was lower than we needed it to be, so we're still undergoing lead optimization. But rather than tell you any more about that, I want to go ahead and tell you about our efforts to identify the target here. So we used, in malaria, it turns out it's been quite effective to generate resistant parasite lines, do whole genome sequencing as a mechanism to finding the target. So this is the strategy that we used, and just to give you a little bit of an idea of how that's done, we started out with 10 to the 9th cloned parasites, and then we treated with 10x EC50 drug in a protocol that Diane Worth, who's in the audience, developed in her lab for a sort of intermittent drug pressure selection method. Parasites clear, then within 48 hours, we wait for them to recrudesce. We isolate the recrudesced parasites. We look to see if they're resistant. If not, we repeat the cycle until we generate a resistant line. And so in this case, it took us 3 cycles, and then we were able to identify resistant parasites in all 4 of the flasks that we had used for these selections, anywhere going from sort of 100 to 1,000-fold levels of resistance in the screen. And then we isolated genomic DNA, sequenced it, and then looked to see what possible information we had about the target. So we actually sequenced 12 clones in the whole genome sequencing experiment, and all 12 of these clones showed mutations in 1 of 2 proteasome subunits. 2 serines were mutated in beta-5 subunit in 9 of those clones, and then there was this arginine 190 mutated in the beta-3 subunit of the proteasome. So it seemed pretty clear from these data that this series is targeting the beta-5 site of the proteasome, which is a known drug target in cancer and also in a number of other infectious diseases. So here we haven't identified new target space, but we've identified a novel chemical series targeting the Plasmodium enzyme. And there was also quite a number of compounds that had been reported on the Plasmodium enzyme, but they had suffered from lack of species selectivity and poor oral bioavailability in that they were mostly peptides that were covalently bound with warheads to the active site threonine. And so this approach then allowed us a fresh start at potentially finding a different binding site to where these inhibitors could be binding. Just to say, we confirmed that these mutations did lead to resistance by using CRISPR-Cas editing to put these mutations back into the wild-type background. And this has been published in this cell chemical biology paper that I've listed. And then we went ahead and purified the proteasome first directly from the parasites after we had engineered in a histidine tag, and then more recently through expression in baculovirus. And you can just see from this table that we've been able to confirm that the IC50 on the parasite, the 3D7, is very similar to the IC50s against the beta-5 subunit of the proteasome, the Plasmodium proteasome, and parallels that for the compounds in the series I've shown here. And then we are also able to show that these compounds are not inhibiting the human beta-5 subunit, so that we really do have a very selective inhibitor series. We went on to solve the cryo-EM structure of the early lead compound 584 bound to the proteasome, and we discovered that it bound to a unique site that's actually 10 angstroms from the catalytic threonine. So it binds here between the beta-6, beta-5, and beta-3 subunits, mostly sort of held in the beta-6 subunit. And if you look here then at the close-in here, it's about 10 angstroms closest contact to the catalytic threonine. So we had identified a pocket that had not been drugged before and really was in a very unusual space in terms of the structure. And so what then was the basis for the species selectivity we're seeing, and that was also very quite evident from the structure. So right here under the circle here, you're seeing that key serine 27, which we saw mutated in one of the strains that helped us identify the proteasome as the target, is actually an alanine in human. And then there's a number of other amino acid changes throughout the pocket. It's actually a quite species variable pocket, including like this methionine to alanine, and then a lot of other changes throughout. So this pocket, which is distant now from the catalytic threonine, and we think is probably represents the S3 to S5 subsites, though we're trying to do experiments to further validate that idea. It really has diverged from that of the human, and that allows us to get this species selectivity. So it's a novel binding site that supports series species selectivity, and we have the series strengths that differ from other compounds that have been reported in that we're reversible, we're drug-like and not being so much a peptide-like structure, this novel binding site. And we did show oral efficacy in an skid mouse model of Plasmodium that I won't show you today. So for this series, then, lead optimization is ongoing, but really shows you the power of starting with a phenotypic approach and working backwards from the target. I next want to move on to a second story, which Jim alluded to, which is our efforts to target dihydroarate dehydrogenase for antimalarial drug discovery. And here in this case, we started with the target, but we knew that from human medicine that this was a druggable target because there's a compound called leflutamide, which is used to treat rheumatoid arthritis. And so here, just again showing our optimization program, and one difference here is we're informing our lead optimization and chemistry right from the beginning with X-ray structures because we knew the target. Just a bit on the DHODH target. This enzyme is essential for the biosynthesis of pyrimidines. It's essential for oxidizing dihydroarate to orotic acid, which then goes on to form the key pyrimidine nucleotides that the cell requires to make DNA and RNA. Now, what's different in Plasmodium is Plasmodium is unable to salvage preformed nucleosides or bases, and so therefore it's completely reliant on this de novo pathway, whereas human cells can actually salvage the preformed bases in nucleosides, so that allows one level of distinction between them. It's a known druggable target. As I said, this compound leflutamide is used to treat rheumatoid arthritis and multiple sclerosis in humans. And it turns out there's a very the binding site where leflutamide binds here, where I've labeled as the target site, adjacent to the flavin cofactor and the orotic acid binding site, is very species variable, and it allows us to then to identify very species selective inhibitors. So again, we started many years ago with a screen of a chemical library, obtained validated hits, and then went through a chemistry lead optimization program. And I'm going to describe to you the two hits that were used to primarily develop all of the chemistry that we've developed over the years. The first one is a triazolyl pyrimidine called DSM-1, and this led to our clinical candidate, which I'll just summarize on the next slide. And the second is this little parole, DSM-43, which has led to our later stage compounds that we're now very excited about that have the potential to move on to preclinical development. So just to summarize the story about DSM-265, this was our clinical candidate that was in the clinics in Peru back in 2008 to 2010. We were able to go from that initial hit, DSM-1, to this compound, DSM-265, which is a potent antimalarial compound with an IC50 on the cells of about 7 nanomolar, fairly similar on the enzyme. This reached phase 2 clinical development. It showed and demonstrated it was capable of a single-dose cure in falciparum patients in Peru. Had a long human half-life of 100 hours, which was something that we'd been driving for in the optimization program. But some of the weaknesses we discovered in the clinic was there was a resistance risk in that we selected for a resistant mutant in a couple of the patients out of just a sort of 30-patient trial. But the program was actually stopped when the organization Medicines for Malaria Venture, who I worked and partnered very closely with throughout the development of this compound, decided we better reposition this compound for chemoprevention as opposed to treatment after they saw the sort of risk of resistance. And we had to do much longer tox studies, and we identified some off-target tox in dogs and a teratogen signal in mice, and for those reasons it was decided to stop development of the compound. But what this compound did do was to clinically validate DHODH as a target for treatment and prevention of malaria. There was also some preventative studies that were run in a challenge mode. It defined the biology associated with DHODH inhibition, demonstrating that these inhibitors have both blood and liver stage activities contributing to either treatment or chemoprevention, and defined a strategy really for positioning these inhibitors in the chemoprevention arm of the portfolio, which has actually gotten a lot more important since the 265 trials ended. So after that, we turned to that parole series that I showed you, and we did get a fairly late lead compound, this DSM-705, and it had some properties that were better than 265 and some that were weaker. It was a bit less potent at 12 nanomolar on the parasite. It did have a reduced resistance risk in vitro assays that I'll be showing you in a minute. It showed better species selectivity versus sort of the preclinical species like mouse, rat, and dog, even though both were equally selective on human. And it had an alternate binding mode, which we kind of attributed to a lot of these better properties in that in this active site of dihydroorotate DHODH, you can see 265 bound in pink, or green, and 705 bound in pink. And you can see that this phenylalanine can form two different positions, and that then dictates whether the binding mode is sort of in this elongated form towards the helix that packs against the membrane or goes up into this other hydrophobic pocket. And we felt that this 705 structure really occupies a much better site and that leads to these better properties. However, the paroles have a very electron-rich ring that has a metabolic liability, so we just couldn't get the needed human half-lives here. So we decided to fix this liability with a scaffold hop. So we worked very closely with Schrödinger, which is a company that has very excellent skills in computational drug design, and we also continued to work with Medicines for Malaria venture. And we decided to engineer a scaffold hop from the parole to the pyrazole. Initial attempts to do that with just a simple pyrazole ring led us to understand we had to methylate the pyrazole, but showed us that we couldn't get out of the ester that you see here in this 679 compound within this little pyrazole to get to an amide that would have been stable. And so we really needed a different strategy. And so we had the X-ray structures of 679, which then could help inform us to how we might make some other modifications that would allow us to get to a sort of stable structure. And so using FEP-guided core hops, Schrödinger was able to come up with about 10 possible bicyclic ring closures and evaluate those with FAP to rank which ones we should make. And we ended up making these pyrazole pyridinone cores and initially identifying DHODH activity at about 1.5 micromolar. So you see we've gone way backwards in potency in terms of the optimization. We're back to really what would sort of be an early hit. But we've solved a lot of the other problems that we had, and we think that this was going to be a scaffold that we could really work on. And that turned out to be true. So just a quick sort of survey in terms of the desired properties. Potency is obviously way off, but in terms of solubility and cytotox and metabolic stability, the core looked like it was a winner, and so we decided to go forward and really optimize this. So this just is going to summarize the entire lead-op program, which is published in this JMedChem paper last year, if anybody's interested. But we worked hand in hand with Schrödinger to use FEP modeling to tell us what we should make those things, test those things, and then solve structures as needed and then come back around. So one of the first key things that was discovered from the modeling was that if we put a methyl or a cyclopropyl in this one position, we ought to get a big potency boost. And this indeed gave us about a 20-fold boost in potency so that we're now down to a 20 nanomolar inhibitor. It was reasonably metabolically stable. And then they decided through the modeling to engage some new backbone interactions with a hydroxyl in the middle that could then form new hydrogen bonding interactions. And this dropped the potency now to under that 10 nanomolar mark. But we were having metabolism issues because probably the hydroxyl itself was vulnerable to metabolism. So we then moved our group, the alkyl group, from this C4 position over onto the bridging carbon, and in that dropped the potency now down to about a nanomolar and a half in this case, and really improved on the metabolic stability. We still had a compound, and this was a really nice little compound, but it wasn't going to be good enough to make it all the way to preclinical development because it still was a little metabolized a little bit too quickly. Actually, its log D was a little too low. And so we did a bunch of med-ID, discovering that this ethyl was a particularly vulnerable site. Once we switched that to cyclopropyl, we really dropped into some nice space where we had very stable compounds, very potent compounds, now sub-nanomolar. And then we were able to explore a few additional scaffolds and finally ended up with this pyrimidine scaffold over here. with a CF3 in the middle as sort of like the ideal compound. And this compound, 1465, is currently in late lead workup with the potential to transition to preclinical development over the summer. It has a predicted human half-life of 130 to 170 hours, so this meets our objective, and all the modeling is telling us that this compound could be a single-dose oral delivered compound for chemoprevention. So I just wanted to say a couple things about confirming the mode of action on this compound. We were able to solve the X-ray structure bound to a close analog and show that we formed the interactions we had hoped to form back with this backbone interaction here. We could demonstrate that we had a good correlation between inhibition on the parasite and inhibition at the enzyme level, really demonstrating that we will likely remain on target throughout the series. But we have an additional trick where we have a cell line that we express the yeast DHODH, which is a soluble form of dihydroorotate dehydrogenase in the parasite. And we can use that to rescue inhibition by compounds in our series. So if we remain on target, this strain should be resistant. And that's indeed what we see in the case of 1465 and other compounds from the series. So we know we're targeting DHODH in our cell killing. And then finally, just to show you a couple, the skid mouse study. This is an in vivo efficacy study that was dosed at 0.1, 0.5, 1, and 3 mg per kg, and we showed very excellent efficacy in this study, actually demonstrating a full cure at 3 mg per kg. And then these data are used and correlated to the plasma levels that provide these various parasite killing activities in vivo to then give us a target value for what we would need to see in humans to actually lead to a cure or prevention in the case of chemopreventative in terms of once you got to humans. So these data are all used to inform then the human dose and half-life predictions. And then finally, in rat PK studies, which have also been paralleled in dogs and mice, we see that we have a very long half-life, very sustained plasma exposure if you look at the plasma concentration versus time profile for this compound. So we have cell killing on target, potent anti-parasitic activity in the skid mouse model, and all of this supports a low human dose of less than 120 mg and a long half-life of over 130 hours. I'm slightly over, but I'm going to just finish in the next couple of minutes. I wanted to tell you a little bit about drug resistance. And so in this case, we turned around a little bit how we did the experiment that I described in the earlier slide. And instead of doing cycling selections, we did continuous drug pressures. And these studies were run in David Fiddock's lab, where we start with variable numbers of parasites at the beginning, and we use that number to help us determine a quantitative assessment of resistance risk. So we're looking to then determine the minimum inoculum of parasites that is required to generate resistance. So we do 3x continuous drug pressure, parasites disappear, and then we wait out between 45 and 60 days for record S parasites. Then we characterize those parasites, evaluating how many parasites it took to get those resistant parasites, looking at the fold change in the potency in the EC50, first in bulk cultures and then in clonal lines. And then we sequence the DHODH gene, or David does, to find out what point mutations or whether there's gene amplifications involved. And what was really interesting to us is we've done this study now over a broad range of compounds in this series. And I've plotted this log MIR, which is the number of parasites it takes to generate resistance at the log form, versus lipophilic ligand efficiency, which is the binding affinity that is not driven by grease, so to speak. So we've subtracted the log D from that. So I'm seeing a correlation between this resistance risk and binding affinity that is not driven by grease. So in other words, the greasier, the more the binding affinity is driven by hydrophobic interactions, the more prone those compounds seem to be developing resistance, and then vice versa. The lower log D compounds are giving you a better resistance profile. And I should note that almost all the compounds on this graph are very, very potent, and so potency alone does not have any correlation. It really is potency driven by non-lipophilic binding properties. And so 1465 here at a MIR of 8.1 easily meets the 7 bar for moving forward in terms of resistance risk. And all of these compounds except this 1463 are better than 265 in the particular selections done under these conditions in parallel. So just a few sort of final other thoughts about it. We're interested in this link between lipophilic ligand deficiency because we think it's a property that you could actually look to optimize during lead optimization programs. But I'd also like to note that there's a lot of literature that says that if you can bind more in the substrate envelope, that puts you in a better space as well. And so we have a structure of coenzyme Q0, which does not have the isoprotenoid tail, but shows you the phenylalanine in the up position, which is where we are in our series. And then on the right here, the Schrodinger is modeled in a coenzyme Q with a two isoprotenoid tail in the tan balls. And you can see that the compound in pink, which is the 1398, the analog of our preclinical or late lead candidate, really fits very well into this substrate envelope, whereas 265 in green is poking out. And so that's a possible other reason for the reduced resistance risk in this series. And so this is an exciting area of research that we're continuing to follow up right now and really trying to understand off rates and entropic versus enthalpic and other energetic contributions. So just to summarize, I think I'll skip sort of that specific compound summary, but just to say that both phenotypic and target-based approaches can identify and exploit key parasite biology for drug discovery. And then finally, I just want to say that you do not do these programs in isolation. These are big team programs, and we had on both programs lots of support from Medicines for Malaria Venture and from collaborators and colleagues in many institutions. Just to hit the highlights, Sue Sharman's group at Monash has done all of our ADME PK. That is, I can't get back there anyway. And then Joe Reedy, who's a UT Southwestern faculty, has worked really hand in hand with me on the proteasome project in terms of the medicinal chemistry. And we've had a lot of support from many people. And sorry for going a few minutes over.